Hello students of class 10. Welcome back to Indrani Ma'am's biology class. In our last class, we have discussed why Mendel has selected garden pea plant for performing his experiment. Today we will know about the process of artificial hybridization. The garden pea plant, is self-pollinating. So, a great care was taken during the experiment to prevent the possibility of self-pollination. How was self-pollination prevented? The anthers were removed from the plants before the stigma was fully mature. The pollens from the parent plant were transferred at the dehiscence stage and were dusted on the feathery stigma of the flowers, from which the anthers were removed. So these cross-pollinated flowers were enclosed in separate paper bags so that further deposition of pollen grains does not take place from other sources. After some days, what happened? The seeds were collected in a bottle and they were studied for observation. The plants which were used as parent plant were said to represent the parental generation. How parental generation was represented? By capital P. And the progeny which was obtained as a result of crossing between the parents of the first filial generation was represented by F1. And after self-fertilization among the F1 plants, the second filial generation which was established was represented by F2 generation. So the crosses which involved the inheritance of only one pair of contrasting traits were known as monohybrid crosses. And those traits with the inheritance of two pairs of contrasting characters were known as dihybrid cross, and those with more than two pairs of contrasting characters were known as polyhybrid crosses. So these three types of crosses has been established by Mendel. Monohybrid cross experiment, what he did, Mendel allowed pea plants to self-pollinate for several generations. And he found that the 7 pairs of contrasting characters he had already identified were already getting transferred from the parents to the offspring. And the seeds which were produced from the tall plants, they produced only tall plants. And the yellow seeds from which the plants were produced, they always produced yellow seeds. And Mendel called each of these pure character as homozygous or pure line plants. And after that, Mendel decided to cross plants with one pair of contrasting traits. Now he selected the tall plants and the dwarf plants, and he made hundreds of crosses by transferring the pollen grains from the tall plants to the stigma of the dwarf plants. And there what happened, when the seeds matured on the short plants, he sowed them again and found that all the plants were tall. And the plant of this generation belonged to the F1 generation. Okay, and these F1 plants were again allowed to self-pollinate, and the seeds again got collected, and after self-pollination, new generation of plants were evolved, and these were the F2 generation or the second filial generation. Now, the plants of this F2 generation were both found tall and dwarf. It had approximate ratio of 3:1. And in the self-pollinating plants, the dwarf plant of the F2 generation produced only dwarf plant. And those tall plants of F2 were found to breed 1:3 ratio, which was found in the previous one. Whether these plants were self-pollinated or cross-pollinated among themselves, they produced in the ratio of 3:1. That is F2 generation. It means that F2 generation contained all the types of the plants: tall homozygous, tall heterozygous, and dwarf homozygous. Understood? So after that, Mendel concluded his studies more further, and he said that the inheritance of character is controlled by the particles, and he called them as hereditary units or factors. The tallness and the dwarfness of these plants were determined by a pair of factors, by a pair of contrasting factors or determiners. Now we term as gene, but previously this term was not prevalent that time. A plant was tall. Why? Because it possessed the factors for tallness. And a plant was short because it possesses the characteristics for becoming short. This led to establish the law of unit character or law of unit factors. Now these determinants occurred in pairs, and these are now known as alleles. Okay, when two factors for alternate expression of a trait are brought together by the process of fertilization, only the dominant character will be prevalent. The factor which is dominant, that will be remaining, and that factor which is not dominant, which is the recessive factor, that will not produce any result. Now on this basis of this type of behavior, the tallness here was described as the dominant and the dwarfness was described as the recessive. Okay, when the hybrids, that is one capital T, one small t, capital T for tallness and small t for dwarfness, these were self-pollinated into two entities and they were united independently. They produced tall plant and dwarf plant. That is known as the law of segregation. Now here from this it is very easy to understand that the one which is dominant character in between the parents, that dominant character will be passing on to the next generation. In the dihybrid cross, what Mendel did, he conducted experiments to study the segregation and transmission of these two pairs of contrasting character at a time. And this cross was known as the dihybrid cross. Now what he did, first basically the parents you have to find out, after that the singular gametes, those gametes will be marched up together and they will produce F1 generation. After that this F1 generation again will be self-fertilization, or we write on selfing. There you have to find out the F2 generation. Okay, now the offspring of the F2 generation which was produced by Mendel, they produced 9:3:3:1 ratio, and he termed those ratio as dihybrid cross. Clear? Now the round characters of the seeds, it will be described as capital R, and the wrinkle character as small r, and the yellow character by capital Y, the green character as small y, and there you have to make out the crosses of these different types of monohybrid and dihybrid cross. Now these genes, how these traits get expressed within an organism. Basically, the genes are the units of inheritance, you know. Where these are located? These are located in the chromosome in a linear fashion, and they are carried from the parents to the offspring by gametes during the sexual reproduction. These control the expression of the characters. The gene works in pairs, and each pair of gene will represent the trait of an organism. That's why your characteristics does not 100% match with your father or mother. Why? Because it has a mixing of the characteristics, it has a mixing of the traits. During the process of reproduction, what happens? The haploid male and the female gametes fuse to form a single cell, and the zygote is formed by the process of fertilization. And the pairs of the genes which is responsible for that particular trait, that will always be present on the corresponding position of the pair of the genes. Understood? And this tallness or dwarfness, that will be decided by the dominant and the recessive character which is prevalent in the parents. Clear? Now a gene, what is it? Basically it is a segment of the DNA with specific nucleotide sequence which has coded information for a specific character, and that specific sequence of nitrogenous base in a gene, it will provide information for the synthesis of a particular protein. The tallness which you have seen in the garden pea plant, that is a characteristic for tallness. The hormones, you know, that is trigger for growth. So the plant height will depend on the amount of the particular hormone which will be synthesized, and the amount of the synthesized hormone, that will depend on the efficiency of the process of making it. Now, if the characteristic of hormone for tallness will give instruction to the plant cells to become tall, then the plant will be tall. And if it is just the opposite, the plant will become short. Understood? And how these blood groups are inherited? The theory is same. Now, human population has 4 types of blood groups: A, B, AB and O. And these are determined by the presence of specific glycoprotein substance which is present on the surface of the RBC, which is known as antigen. The person of blood group A will have antigen A. The person of blood group B will have antigen B. The person with blood group AB will have antigen A and B. And the person with blood group O does not have any type of antigen. Understood? So based on that, the blood grouping will be divided. 4 phenotypes are present. Blood group, we write capital A to the power, as you write, it will be AB, AB and O, 4 separate. So you have to make a cross. Now, if a homozygous blood group B man marries a woman with blood group O or vice versa, how will the children be produced? The children will have blood group B. You have to show it. You have to make the chart of the cross. Second criteria, if a man who is having blood group A will marry a homozygous blood group B woman, the blood group of the children will be AB. You have to show how this is formed. If a man heterozygous for blood group A will marry a woman who is having blood group B, how will their children be? All will have chances of 25% AB. There will be a 25% A, 25% chance of having B and same 25% chance of having O. And if a man with blood group AB marries to a woman who is having blood group AB, their children may have blood group A, B or AB. How this is forming? There is 50% and 25% chances of having all this. Understood? Now, how is the sex of a child determined? The sex is the hereditary difference between 2 individuals of the same species. Sex is the most conspicuous and interesting character of hereditary differences observed among the individuals. The process by which the sex of a person is determined is called sex determination. In unisexual animals, including human beings, the chromosomes are of 2 types. One is the autosome, another is the sex chromosome. The autosomes, these are basically found in the somatic cells, and these autosomes do not determine the sex of the individual. They determine only the somatic characters. Somatic characters means the physical body characters, and these we write as capital A. Understood? And sex chromosomes are found in the sex cells, and these are responsible for determination of sex. X and Y chromosomes are present. The female is having XX chromosome and the male is having XY chromosome. So in human beings, these two, the female can only give the X chromosome, whereas the male can give either X or Y chromosome. If the X chromosome from the male fuses with the X chromosome of the female, then a girl child will be formed. And if the Y chromosome from the male fuses with the X chromosome of a female, then a boy child will be formed. Now the sex of a child, what type of child will be produced, is a boy child or a girl child, that entirely depends on the sex, which is sex chromosome, which is liberated by the male. Understood? Sometimes sex determination is regulated by environmental factors also. In some reptiles, the temperature in which the fertilized egg is incubated, that will determine, before hatching, that will determine the sex of the this type of organism. That will determine the sex of this reptile. In case of lizard, high incubation temperature result in the male progeny, while in case of a turtle, high temperature result in the development of the female progeny. Understood? So till this much today, we will continue the remaining portion in our next class. Thank you.